Okay, we're going to get going. We have the chair of the Cancer Redox Biology faculty again, David Wink, talking about biochemistry.
Terry, and yes, you two can be bored for an hour, but Dave, I promise you, Dave Roberts, you guys will have a lot more fun with what he has to tell us today. So what I'd like to do today is we're going to continue with what we were doing with chemistry, but we're going to talk about some of the biochemistry, some of the ways that redox molecules are formed biochemically, and some of the enzymes are familiar again with the nomenclature, so that you actually have a vocabulary to read read redox biology. I apologize for the ragweed. It's been really bad on me. For some reason, I never had this problem until I came to this area. Hopefully I'll get through it. So one of the things that we've been talking about here is the instigators. These are the things like the hydrogen peroxide, nitric oxide, as Terry mentioned, carbon monoxide, even hydrogen sulfide, which I'm not going to talk too much about today. But the sources of these, a lot of them, some of these are endogenous radicals, but some of the sources, like for ROS, is NADPH oxidases, the P450 flavins, xanthine oxidase, mitochondria, nitric oxide synthase. This is one of the primary sources for nitric oxide. And heme oxygenase is one of the primary sources for carbon monoxide and bilirubin. Like I said, I won't talk too much about hydrogen sulfide, but there's a whole series of enzymes, and I probably should put that in next time. Oh, you're going to do that. Okay, great. Did you get my P&S paper? But on the other side, we can form these radicals in nitric oxide, such as nitric oxide, and we have to have ways of detoxifying some of these species. Things like superoxide dismutase, catalase, which scavenges hydrogen peroxide. If you ever get a hydrogen peroxide sensitive electrode, it's kind of fun to put like a millimolar hydrogen peroxide, put a tiny bit, a micromolar amount of catalase, and it just consumes it so rapidly. So it gives you kind of a sense of how biologically we can consume things like hydrogen peroxide. DT diapherase, these are 2-electron processes that can undergo scavenging. Glutathione, ascorbate, and tocopherol, or the vitamin E. So, what are these sources of instigators that generate hydrogen peroxide and superoxide? This is N8-NOx, or NADPH oxidase. Tom Lado will be talking about this. This is a very important enzyme, both in signaling and in antipathogen. This is xanthine oxidase. And xanthine oxidase, every couple of decades, gets reinvented as something else. Recently, people have talked about xanthine oxidase is reducing nitrite to NO under hypoxia. And that's important because if you're under hypoxia, nitric oxide dilates vessels to get oxygen back to the tissue. This is an overlooked enzyme, but it's becoming more and more important in different diseases, polyamine oxidases. And these are associated with arginase. Arginase, if people are familiar with, it's like Th2 response. And these, from the polyamine oxidase, we can generate hydrogen peroxide. And from hydrogen peroxide, of course, we can then end up with hydroxylated lipids, oxidized proteins, and of course, oxidized thiols. So there is an antioxidant system. So if we have, this is pro-oxidation, we have an antioxidant system. Catalase directly scavenges hydrogen peroxide. We'll talk a little bit about the chemistry. Peroxyredoxins also will scavenge hydrogen peroxide and also lipid hydroperoxides as well. SOD, superoxide dismutase, prevents superoxide from actually donating electrons to metals. We talked about the Fenton reaction in Haber-Weiss last week to generate things like hydroxyl radical or iron oxo species. And of course, glutathione. And I can't overemphasize the importance of glutathione. If you want to test this, treat cells with hydrogen, tumor cells are really good. Treat cells with hydrogen peroxide and then use things like BSO to deplete the glutathione. You see what a tremendous increase in cytotoxicity. Glutathione has been adapted in cells as a universal detoxifying enzyme, or I'm sorry, peptide. And then finally, these oxidized proteins here One of the antioxidants is thioredoxin. If we actually look at nitric oxide synthase, this is another instigator here. We can generate nitric oxide. It turns out superoxide and nitric oxide can annihilate each other, and this can be regulated by NO. But actually, NO has another important function. This is one that we actually discovered back in the early 1990s. Nitric oxide, especially at lower doses, is a very powerful antioxidant. People kind of overlook this because they're always associating nitric oxide with all these components of air pollution. But nitric oxide is a very powerful antioxidant. And how nitric oxide is really controlled is really two ways. One is cellular consumption. And this isn't really well defined yet. Some people think it's through the mitochondria. Other people have shown that it's through globins within the cell. And also hemoglobin. Red blood cells do a wonderful job of scavenging nitric oxide, and they convert it to nitrate. And we'll talk about that today. So let's talk about the outer sphere electron transfer here. If we have electron transfer directly to oxygen to make superoxide here, this is upstream. This is increase in thermodynamic energy here. If you protonate this, it actually increases its reduction potential here. If superoxide then is another electron's added, this is a downhill reaction, and this makes hydrogen peroxide. So there's always the adage that when you generate superoxide, a lot of people talk about, oh, superoxide's a bad player here. But often people overlook the fact that superoxide dismutates to hydrogen peroxide. And this is actually a very important driver of a lot of the oxidation reactions. So if you have things like paraquat, if it's reduced, can actually reduce O2 minus here. And again, we go through these equations here that this is a downhill reaction to oxidize methyl viologen here. And even though this is an uphill reaction, the downhill reaction here is favored by 0.12 volts, therefore it's spontaneous. So you can actually reduce things like paraquat, and you put it in solution, and you can generate superoxide. Oh, by the way, if there's anybody who wants to stop me, that's no problem. Dave doesn't talk till 5 o'clock anyway, so we can talk about that. But one of the things about this redox cycling, these are xenobiotics, and many compounds can actually donate electrons to oxygen. This is through this outer sphere electron transfer. Ubiquinone, menadione, or vitamin K, adriamycin, or doxorubicin, which is a chemotherapy. And these are a class of compounds, the quinones here. And some of the quinones can take an electron, take an electron to form semiquinones. And semiquinones have unique properties. One of the properties is it can donate an electron back to oxygen here. If you make a semiquinone here, it can actually go on to form a diphenyl complex here. But this semiquinone can also bind to things like glutathione or bind to proteins, sulfurs and proteins here. And this is one of the mechanisms how semiquinones can actually be toxic beyond just generating hydrogen peroxide. Dopamine is one of the classic examples here, and some of the estrogens here. This is an orthoquinone here. And when you make this semiquinone here, this can be then converted to the orthoquinone, the diphenol here. And this is like adrenaline. And you can actually generate from this, if you act from the semiquinone here, you can actually generate here superoxide as well. So even though you have things like norepinephrine or dopamine, you can actually undergo the quinones, and depending on the electron potential here, you can actually generate things like hydrogen peroxide, or some of these can actually bind to some of these thiols. Menadione, or vitamin K here, this can actually oxidize, its oxidation potential, it can actually oxidize hemoglobin. Hemoglobin is in a ferrous state, or iron 2 state, which then can go to methemoglobin, which is an iron 3 state, and form the semiquinone, which can result both in superoxide or react with thiols. Here again, dopamine here. Reduced dopamine is in the diphenol state. And when it gets oxidized, it can form 6-hydroxydopamine. And this here can actually be a potent toxin, or neurotoxin, and actually lead to things like Parkinson's disease. So pro-oxidant versus antioxidant. So if you can put an electron in or you can take an electron out, you don't know whether you're driving oxidation or you're protecting against it. In this case here, we have quinones. You can form these semiquinones here. The semiquinone has the option, again, if there's enough oxygen around, or it does something else. And when we talked about the Fenton reaction, the Fenton reaction reacts with hydrogen peroxide plus iron 2 complexes. So we can actually reduce this and make this a pro-oxidant. So if you make the semiquinone here and you form superoxide or you reduce iron here, you actually have a pro-oxidant. This can actually result in driving or catalyzing the reaction of the Fenton reaction. The antioxidant role reduces the semiquinone to actually diphenol. And a lot of the reactions that we'll show you, especially with things like vitamin E, you actually want to go to the fully reduced form, that the downhill reactions end up at the fully reduced form. So one of the things about when you're trying to decide whether something is going to have pro-oxidation or antioxidation, location and pharmacokinetic stability, like water versus fat, these are actually very important factors in determining how these things work. So this is one of the things with vitamin E. Vitamin E is tocopherol here. And tocopherol can be oxidized by a variety of different powerful oxidants. Lipid hydroperoxide, hydroxyl radicals, NO2, nitrogen dioxide. Remember, nitrogen dioxide's the brown gas that's over Los Angeles. Terry knows that all too well, that kind of thing that works up the valley there. But NO2 is not very healthy, and things like vitamin E can be oxidized here. But one thing about vitamin E, if under the right conditions, it can actually start donating electrons to further drive oxidation. The Fenton. Does everybody remember when we talked about the Fenton reaction last week? But this also can react with ascorbic acid, so vitamin C. And the vitamin C can actually convert this back to tocopherol there, and then the ascorbic acid, of course, gets further oxidized and gets recycled here with ascorbic acid. But then this cycle here with ascorbic acid actually now becomes detoxifying. Carotenoids. You have the very same thing here. You have alkene groups in a very long chain here. And again, hydroxyl radical or lipid hydroperoxide radicals, they can actually oxidize this, form these lower radicals. So one of the important things when you're talking about antioxidants, like in vegetables or different types of products, is when you have a powerful oxidant here, it reacts with it to form something that has much lower potential. That's what you want to do. If you react with something that stays up here, the potential, then it can actually oxidize other substrates. So things like vitamin E, the carotenoids, things that your mother told you to take, which I don't do anymore, and that's why I look like I do, you know. So you have to have things that react and then can go to much lower energy. So xenobiotic metabolism. This is the classic P450s. Things like quinones or paraquats can be oxidized to these, in some cases, semiquinones. And one thing that radicals, high energy radicals, Mother Nature does not like to do this. Okay, one thing that Mother Nature does, we talked about outer sphere and inner sphere electron transfer. In nature, we don't like to have unaccounted electrons unless it's on purpose. Nitric oxide actually is one of those things that's on purpose. At least I think so. So in this case here, we have a condition here where we generate this, and again, it can drive oxidation reactions here. But there is an enzyme, quinone reductase. This is NQ1. If you actually look at things like the antioxidant responsive element, sometimes if you expose cells to stress, you upregulate a host of defense mechanisms. They usually have what they call the ARE promoter site, and it's called the antioxidant responsive element. One of the big ones is NQ1, which is part of a P450. But instead of taking a substrate like X, instead of generating this kind of radical, which can go either pro-oxidant and have other types of chemistry, this actually directly, with a 2-electron transfer here, reduces it to the dihydro. Things like broccoli. If you take broccoli, induce this ARE. There's the ADTs in there, or the some of the thiol compounds do. So now let's talk a little bit. Am I going a little too fast? If I am, please slow me down. The mitochondrial sources of reactive oxygen species. How many times have people read that ROS coming from the mitochondria is killing the cell? Dave's read that. You've read that. So where do they come from? Now, I'm not a firm believer there's a lot of hydrogen peroxide that comes out of the mitochondria, but ROS coming out of the mitochondria is, if nothing else, important to signaling. So as we took when we designed this course about a decade ago, we used to talk about toxicity. Well, we've become a little wiser as we've actually done our own research. And you have to start thinking these things in terms of signaling first. Then we think about ROSs in terms of actually killing the cell through just deleterious mechanisms. So in the mitochondria, we have site 1 here, which is generally a site where NADH will bind and transfer electrons to site 2. And in site 3, oxygen, interestingly enough, can be converted to O2 minus. So if the electron that sits on site 1 isn't oxidized by cytochrome C fast enough, then oxygen can come in and generate superoxide here. And then finally, the electrons end up in site 4. Oxygen comes in, binds to an iron complex, which we'll talk about in a minute, and then is converted to water. So this is your basic mechanism. Most of the ROS stem from this site 3. So in site 3, there is a quinone, and just like some of the compounds that we were talking about. So there's ubiquinone, and ubiquinone can actually form a semiquinone. So if one electron comes down and reduces ubiquinone to the semiquinone, then the semiquinone can actually reduce oxygen back to O2 minus. And depending on the pH, because the pH, depending on the proton pump, can change the pH in different parts of the mitochondria to up to 4.7. So that makes actually superoxide even a more powerful reductant. But ubiquinone can also form ubiphenol. And so if the electron comes in and it doesn't react with oxygen, it can actually be re-reduced to ubiphenol here, which actually is a much lower potential. So the polarization and depolarization of the mitochondria can affect the reduction potential here of the proton and where the proton pump is. So when you talk about if you poison the mitochondria, you can actually look at, if you've ever done this, you guys might have already done this, you can actually look at with specific dyes the polarization of the mitochondria. So some of the energetics about this, about electron transfer, is that in the case here where we have the quinone and we have superoxide, now superoxide could react with peroxynitrite, but one of the things that manganese SOD, that's the mitochondrial specific SOD, resides there in quite high concentration, and it reacts with superoxide near diffusion control. And one of the things that it can do, if the manganese SOD is high enough, it converts superoxide instead of superoxide reacting with the quinone to form the semiquinone. What ends up happening is manganese SOD can actually generate hydrogen peroxide. And the hydrogen peroxide, often generated from manganese SOD, can light up dyes such as dihydrorhodamine and DCHF. So this balance here between the quinone state, superoxide, and the semiquinone here, people have always talked about this is the source of the ROS. But way back when, Bruce Ames, I think he sat in a TV show because of this reaction, and he said, we're being irradiated all the time because we're generating all these radicals and hydrogen peroxide, and that's why we're aging. I'm not quite sure if this is true. There's been recent reevaluation of this, though you can get this here. This equilibrium lies strongly in favor of the semiquinone and not in favor of the O2 minus here. So let's talk about other oxidation chemistry that's important for instance, in cancer. Bleomycin, which is a chemotherapeutic drug, it's an iron complex here. And we talked about iron in the Fenton reaction last week. If you take iron and you put an electron in, you form the ferrous compound here. And the ferrous compound can actually react with oxygen and pick up another electron. And when this happens here, this is going to be a common mechanism that we're going to see, is this reaction between oxygen and these reduced ferrous compounds here. Then you can form these powerful oxidants. These are iron oxo species. And they're just almost as powerful as the hydroxyl radical. In this case here, when we form that, we can actually oxidize things like DNA. And in some cases here, we can generate things like equivalent to hydroxyl radical or the iron oxo species. And when we undergo substrate oxidation here, we regenerate this. And because we use one electron cycle here, we can actually develop a cycle here which will actually destroy DNA. But on the other side, so we've shown how this can generate a powerful oxidant here. The same chemistry can be applied to prolohydroxylase. And why is prolohydroxylase important? If people are familiar with HIF-1 alpha stability or collagen, scurvy, right? Everybody remembers reading about scurvy? Well, prolohydroxylase is essential for stiffening up, oxidizing the prolines to stiffen the collagen up. If you don't have enough ascorbic acid, which is your electron donor here, then you get scurvy because your collagen is kind of wobbly. Is that right? I think that's how the best way to describe it. I remember learning that in 6th grade, so that's why it stuck. So if you have this iron 3 complex, just like here, iron 3 takes an electron, generally from ascorbic acid, reacts with oxygen. Again, we get these other high-valent iron complexes, and we the hydroxylation of proline. If it's in the HIF-1 alpha, then in fact it sets HIF-1 alpha up for degradation to E3 ligase. So the same chemistry that we use to kill and cleave DNA can also be used in signaling and building the cell. So P450s are a really important family, and I don't think they've talked a lot about it, but when I was going through school, P450s were all the rage. Things like how drugs were metabolized, how carcinogens were made. P450 is a ubiquitous heme family, which actually does a lot of unique oxidation chemistry. And how it happens is, again, you have this resting iron state here. You get an electron from the P450 reductases to form an iron 2 complexes. Once you do this, just like in hemoglobin, it binds oxygen. Another electron's put in, and you actually form this superoxide ferric complex, which then gives off water and forms this high-valent iron complex, which actually has an electron oxidation potential of 1.4 to 1.6. And then you oxidize through the substrate here, and you return to resting, and then you have this catalytic cycle. The P450 reductase that donates this electron here comes from NADPH through a flavin, and if the flavins don't transfer the electron to this iron complexes, it decouples. Things like quinones and even oxygen can form these semiquinones and generate O2 and also hydrogen peroxide. And this is called the decoupling mechanism. Often people used to observe the P450s when they were elevated and they did not have substrate, or this iron was actually poisoned. It would uncouple the enzyme and generate a lot of ROS. So what are some of the reasons we purposely want to make reactive oxygen species? One, we want to form oxidation. One of the purposes is antimicrobial. It's hydrogen peroxide and all the derivatives of hydrogen peroxide is kind of essential for fighting pathogens. It's also a product of metabolism, and it's also part of signal transduction. So these are some of the enzymes that generate hydrogen peroxide. Glucose oxidases, which is primarily bacterial, generates from glucose hydrogen peroxide. Oxygen plus spermine and polyamine oxidases generate putrescine and peroxide. Xanthine oxidase, which we use it a lot actually to generate both superoxide and hydrogen peroxide, can convert oxygen into urate, superoxide, and or hydrogen peroxide. Hypoxanthine can also be used, and this is one of the substrates. If you're going to use this enzyme, this actually has a higher affinity than xanthine, and the rate to make xanthine, you generate hydrogen peroxide and O2 minus actually 7 times faster with hypoxanthine. And NADPH oxidase, and you'll hear a lot of later on in the course from Tom Lato and all the regulation, is NADPH plus oxygen generates 2 superoxides. So xanthine oxidase, one of the purposes of the xanthine family is that for purine metabolism, it detoxifies aldehydes, and it's also the generation of ROS. Now, xanthine dehydrogenase basically converts xanthine, part of the purine metabolism, to uric acid and NADH. So the dehydrogenase form, which is actually the first form that's made when you're synthesizing this, actually ends up generating NADH, but not O2 minus, superoxide or hydrogen peroxide. But in the case of disease or injury, especially under hypoxia or when cells are stimulated with cytokines, xanthine dehydrogenase can be clipped and form xanthine oxidase, which generates a hydrogen peroxide and H2O2. One of the classic examples, Neil Granger, I remember him back in the 1990s describing a lot of this chemistry with Matt Grisham, and they were talking a lot about this in ischemia reperfusion injury. And so xanthine oxidase is a unique kind of enzyme. It's a molybdenum-based enzyme. We've been talking mostly about iron and maybe iron and copper enzymes. But in this case here, your hypoxanthine and xanthine actually is metabolizes to urate, giving its electrons to molybdenum. Molybdenum passes it on to the FAD, and then the FAD H2 plus actually is then oxidized by oxygen to either O2 minus or H2O2. And the ratio of superoxide to H2O2 is actually pH dependent. It's actually temperature dependent. It turns out hypoxanthine will generate a little more superoxide, but xanthine actually produces more hydrogen peroxide. So you can you're actually generating both of these when you actually use xanthine oxidase. It turns out mouse, I thought this was kind of a neat fact, mouse utilizes xanthine oxidase much more than humans. And this molybdenum, this unique molybdenum, there's a whole bunch of different enzymes that are molybdenum-based. Sulfide reductase, for instance, both mammalian and bacteria, have this molybdenum cofactor here, which is bound to this moiety through these thiol groups here. And it can actually oxidize here xanthine and hypoxanthine. One of the most important things, too, is allopurinol. Here, you can actually give this systemically. Will inhibit this enzyme by binding to the molybdenum group. So NADPH oxidases, again, you'll get a much in-depth lecture about this from Tom Lato. There are several isoforms of this, but it's a highly regulated enzyme here. One of the phagocytic functions of it is it's antimicrobial. And if a monocyte or macrophage sees an organism, it will actually engulf it in the phagosome. And one of the proteins that actually gets transferred to the phagosome is NADPH oxidase. The other thing it can do, though, is this is mostly what they call NOx2. NOx1, we'll talk about NOx1, 3, 4, and 5, actually aren't really antimicrobial. They regulate cell surface signaling and regulation of a number of different physiological functions. But NADPH oxidase consists of a bunch of different players. The GP91 FOX, we'll talk about this. This is the part of the protein that actually the oxygen gets reduced to superoxide and hydrogen peroxide. And then there are regulatory, cytosolic regulatory factors, P47, P67, and P40. And also RAC and RAC2 play critical role in a lot of the different NOxes. So what does this look like? Actually, it's kind of an interesting enzyme. These are heme proteins that are deep in the pocket here. And what they do is when they're reduced from NADPH to FAD, the ions get reduced. And this is in the GP91 FOX domain. And where oxygen then, through an outer sphere electron transfer, forms superoxide. And this is the cell one. If you ever take an activated neutrophil, murine activated neutrophil, they produce an impressive amount of superoxide. You can actually measure this. And they can produce up to, as I remember, 10 to the 6 cells, they can produce like 100 micromolar equivalent of hydrogen peroxide or superoxide in an hour. So that's quite a bit of superoxide. But to get this enzyme to actually work, it's critical that all these factors have to come together and get phosphorylated. So it's an incredibly highly regulated enzyme. The GP91 is where the business end, and the 67, 47, and 40 in the rack are actually critical here for the activation and the flow of the electrons. So nitric oxide synthase, this is one of my favorite enzymes. Nitric oxide synthase, we've talked about these heme proteins. A lot of them generate powerful oxidants. The P450s, we haven't gotten to the peroxidases, but in this case here, nitric oxide synthase is also a P450. Is everybody familiar when I say P450? They usually have a heme site, they have a thiol on the distal side, and then you have the active part here where the oxidation occurs. And so nitric oxide synthase are two types. One is the inducible form, and the other is constitutive. In the constitutive, this is NOS1, or neuronal NOS. It was first discovered by David Brett and sequenced by David Brett in Sal Snyder's group. NOS3, eNOS, was actually the nitric oxide synthase, I think, was first described, and they gave the Nobel Prize out for the production of nitric oxide and EDRF. But the inducible nitric oxide synthase, this generally is not constitutive. If you take eNOS out, you get hypertension. If you take eNOS knockout mice, their systemic blood pressure actually goes up. nNOS knockouts, we're not quite sure what they do. They do have some dysfunction in their GI tract. But the inducible nitric oxide synthase actually comes up, and in murine models, it primarily comes up when you activate things like monocytes with LPS. The nitric oxide synthase have the P450 reductase domain. And unlike P450s, where P450 can be here and it has to bind to its reductase domain, the nitric oxide synthase actually has, the whole protein has the reductase domain together. That's one of the unique things about nitric oxide synthase. So it has this reductase domain, so it can accept NADPH electrons. It's cal module binding, which is very important because it's calcium dependent. These two are calcium dependent, and NOS2 is actually calcium insensitive. In this case here, we have heme oxygenase domain, and this, like P450, when you reduce it, you combine oxygen, and it converts arginine to N-hydroxyarginine, which is ultimately oxidized to NO. And there's a lot of things, like we were talking about the NOx system. A lot of factors control this. In fact, as somebody told me this, that nitric oxide synthase is one of the most highly controlled enzymes at all levels, not only at signaling levels, also what type, the protein level, and the expression level, but also it's intertwined with a lot of metabolism as well. So in the nitric oxide synthase, the way that it really works, it only works as a dimer. So you have a monomer which has a heme oxygenase domain and a reductase domain. You have tetrahydrobiopterins, which actually sit between these. But instead of the electrons going from in the monomer to the heme, it actually has to bind head to tail with another dimer to actually become active. So the electrons flow from this reductase domain to the heme and vice versa. So the dimerization is actually very important. Dennis Stewart and his group described this years ago. So, so this monomer to dimer ratio is actually very important to understand the activity. And again, arginine is converted to citrulline and nitric oxide. So some of the biology we're describing is that it's dependent on arginine. So it's linked into, it's actually in competition with enzymes such as arginase. Oxygen, it's oxygen dependent, NADPH dependent. It's the calcium dependent. Now this is, the calcium dependency is only with the nNOS and the eNOS, not with the iNOS. Does that make sense? So one of the best examples is acetylcholine binds to the endothelial cells, or bradykinases. Like you get the calcium influx in there, it turns on eNOS, and you get vasodilation. But in the case of the iNOS, which produces probably 10 to 100 times more nitric oxide per cell, it's calcium independent. And from nitric oxide, you get nitric oxide, of course, you can measure that. NOAA, you actually get this hydroxylated arginine. We used to think it was just a substrate, but NOAA actually has another function. It inhibits the arginase pathway. An arginase pathway is very important because arginase, if you have a proliferating cell, it requires the conversion of arginase to polyamides to actually induce proliferation. So NOAA actually can inhibit the arginase pathway. If it's uncoupled, it can generate both peroxide and superoxide. And there are some people that think that nitric oxide synthase, under some conditions, can actually produce the nitroxyl, which is a reduced form and has its own unique biology from nitric oxide itself, just by one electron here. So the iron nitric oxide, one of the things that was very interesting that Dennis Stewart's group showed was you have this equilibrium. If NO is bound to the iron, it can come off here, and it forms this equilibrium here. And what he discovered here was that each one of these KIs was very different here. So if the KI for inhibition of nitric oxide was high for eNOS and nNOS, that means these two enzymes here don't produce so much NO because the NO lies on this side. And the iron nitrosyl complex can't cycle around to make more nitric oxide. But iNOS, interestingly enough, Ki was much, I think I have that backwards, lower. And in fact is that the Ki, I'm sorry, the Ki is measured this way. In this case here, the inhibition here of nitric oxide allows iNOS to actually release its NO much higher. And also the Km for oxygen here at very low concentrations, such as about 20, the oxygen concentration, the Km here, also differs between each one of these. This is about 22 torr for eNOS. This is about 100 torr or so. And nNOS is actually very, very high, has a very high Km, almost 800 torr. So you have different selectivities for different isoforms, and that's why I think you have all three of these, because they do different functions. You have the low affinity for oxygen to induce turnover. That's because many times when you're controlling blood vessels, you actually get a little hypoxic, and you can still generate nitric oxide. But nNOS is more controlled, and it requires a higher, oxygen content here. But nNOS is very interesting because if you looked at the physiological pattern of oxygen, it's actually a straight line under the physiological. The dependency on oxygen is actually a straight line through the physiological concentrations of oxygen. And it was postulated that nNOS may actually be an oxygen sensor in some ways. So we're going to keep moving on. Most of these enzymes we're talking about now are P450-like. This is heme oxygenase, and it there's two isoforms, heme oxygenase 1 and heme oxygenase 2. And what they do is when you have free heme, they convert heme into biliverdin and ultimately to bilirubin here, and it generates carbon monoxide. And it turns out that carbon monoxide is an interesting gas. Many people have actually shown that carbon monoxide, breathing carbon monoxide, can actually be immunosuppressive. There's a group in Pittsburgh, when they first described this, would actually have people in septic shock breathing low doses of carbon monoxide, and they ameliorate the septic shock. But you can produce CO, but you also produce iron, but you also produce bilirubin, which is actually an antioxidant. So we talked about how we induce, how we actually generate these different types of radical systems here. But there's also where we are inducing NO or peroxide, you also have to have systems that actually detoxify it. So one of the things that we do is if you've generated a powerful oxidant or an NO, you need to scavenge it. And one of the ways that you scavenge is directly having a molecule bind to it, like ascorbic acid. It's an antioxidant. We have this in our shampoos. And again, what are the requirements of having a quenching reaction? You want to have the products, ultimate products that are scavenging these oxidants be innocuous. A lot of them, it'd be nice to have them biologically recycled, and also that they prevent any chain reactions that may occur through the oxidation process. So if we have here some of the biochemistry, we go back to here, we generate O2 minus and hydrogen peroxide. Hydrogen peroxide can bind to iron 2 and ultimately end up with lipid peroxidation. Hydrogen peroxide can react with peroxidases. We'll talk about those in a little. Again, these are heme proteins here. They can make HOCO. This is basically the chlorine that you have in your pool, hypochlorous acid. You also can react with bromine. All these have antimicrobial properties or NO2, nitrite reaction here. But hydrogen peroxide, in addition to being antimicrobial and playing a role in a number of generation of a number of different reactive species, can also inhibit things like aconitase. Your TCA cycle, your citric acid cycle can be inhibited here. Hydrogen peroxide can actually react with things like cyclooxygenase or lipoxygenases to activate those. Those are your carcinoid pathways. And H2O2 can actually modulate MAP kinase pathways, NF-kappa B, and even P53 pathways. A lot of that's through thiol metabolism. Nitric oxide, if we produce nitric oxide here, nitric oxide is being produced, and we developed this thing called the chemical biology of NO, trying to sort out all the different reactions that are possible with nitric oxide in a biological system. It's kind of daunting. So back in the 1990s, we decided that to try to catalog what we think is probable. Like we can have reactions that occur at 3 atmospheres. You know, it works fine on Venus, but it does not really work well in physiological conditions. So in this case here, we have nitric oxide synthase, and we make NO. NO, these reactions with guanylyl cyclase that regulate blood pressure, this is actually a very fast reaction, require nanomolar levels of nitric oxide. NO can inhibit P450 because it can bind directly to the heme, or it can scavenge radicals and have antioxidant properties. But when the levels of nitric oxide start to increase, nitric oxide can also be an antipathogen as well. It's part of the arsenal, antipathogen arsenal. And when macrophages use that, you can generate these intermediates here, NO2, N2O3, resulting in oxidation nitration or nitrisation. And these species here can have anti-tumor and antipathogen activity. Another mechanism which is very important in detoxification is controlling superoxide. Again, we don't want superoxide just simply to run around and reduce things. Aconitase, for instance, is actually reduced by superoxide. So we want the rapid conversion of SOD to hydrogen peroxide. But by converting it to hydrogen peroxide, there's a lot of enzymes that control the concentration of hydrogen peroxides. Catalase is one of the big heme proteins that do it. The other one that's constitutive, catalase generally is upregulated, is glutathione peroxidase. And unlike the catalase, which is a heme protein that we've been talking about, it's actually a selenium compound. And it's both cytosolic and mitochondrial. And the GPX plays a key role in detoxifying most of the cells hydrogen peroxide under normal conditions. When we get these pathogenic increases of hydrogen peroxide, then we actually increase catalase activity. Another way that hydrogen peroxide is detoxified, again, is through the peroxyreductants. Sugu Ree, when he was here, I think in this building here, was describing a lot of this chemistry of the peroxyreductants here. And there are several isoforms. There are 6 of them that I know of. They either have 2 cysteines, 5 and 6 actually have 1 cysteine. And like the selenium proteins, they directly react with hydrogen peroxide or these lipid hydroperoxides and detoxify them. And one of the important things is where the location of peroxyreductants are, and they can actually control signaling. So one of the important things that peroxyreductants, yes, they can react with hydrogen peroxide or lipid hydroperoxides, but they're usually in places where they're trying to control oxidative signaling as opposed to oxidative or just simply detoxifying hydrogen peroxide. Scavenging of nitric oxide, that's really important. And nitric oxide can be scavenged by globins. This is a very fast reaction. Cytoglobins that are actually inside the cell itself can scavenge nitric oxide. But when nitric oxide is diffusing through tissue, one of the biggest sinks for nitric oxide is actually the red blood cells. It never really survives in the blood for very long. I think it's on the order of microseconds in the blood, and it never really reaches high enough concentrations to actually or long enough lived or high enough concentration. So hemoglobin plays a critical role in controlling nitric oxide and locating it right to the cells that it's being produced in the tissue. And also nitric oxide, one of the things that if you actually, well, I don't know if you have the apparatus, but if you actually have a nitric oxide electrode, you can actually put like a million cells in a dish, and you can watch a micromolar of nitric oxide actually be consumed. There's a lot of papers that show this too as well. So cells themselves can actually consume nitric oxide. And there's a question as whether it's actually mitochondrial or some other protein. Another important mechanism, though, is these iron oxidase complexes. We talked about the antioxidant effect, but these reactions are also very fast. So if you're generating a lot of hydrogen peroxide and they're reacting with these metals here, the nitric oxide will actually convert those metals from these high oxo metals actually into benign iron-3 complexes. And it's a critical way not only of nitric oxide being an antioxidant, but also controls, it's critical in controlling the NO. So another system here we talked about was the glutathione systems here. And glutathione is one of the front-end mechanisms for just about anything from nitrosative stress to oxidative stress. And glutathione reductase is critical. If glutathione gets oxidized to form the disulfide, it has to be re-reduced back. But what ends up happening is your glutathione, your oxidized glutathione will start building up in the cell. And that induces oxidative stress. And so, how this occurs here is GPX is, in this case here, glutathione can be oxidized to GSSG. In the case of GPX, it's a selenoprotein. So the selenoprotein will bind to hydrogen peroxide. It will actually scavenge things like lipid hydroperoxides. And what happens is these actually directly react with the selenium here. And they form these selenium hydroxide adducts here, which in the presence of GSH go to water. And then this protein here, the selenium thiol adduct here, then reacts with GSH here and then reconstitutes the protein. And you form this catalytic cycle that actually can detoxify things like hydrogen peroxide. And it's critical, the functionality of this glutathione reductase here. There's other selenium compounds, epsilon. You can actually buy this. It's a small compound that people have tried to promote for good health because it's a selenium-based compound. So let's talk about peroxyreductants. Many of them are overexpressed in breast cancer, liver cancer, prostate, oral, and lung. And they do play a role. The isoforms 1 through 4, they have this disulfide. Again, they react with hydrogen peroxide directly with the thiol. They form a disulfide, and they reduce hydrogen peroxide to water. When you make this disulfide in a protein, the glutathione reductase can't actually re-reduce it. There's another very important protein, which is called thioredoxin. And thioredoxin will actually react with this, restore this disulfide bond, form a disulfide bond in itself, and then is reduced by another protein called thioredoxin reductase. The thioredoxin reductase is also a selenium protein. So selenium has a critical role in these antioxidants mechanisms here. And you take NADPH, and again, through this flavin transfer here, you reduce the SS bond back into the thiol. And so you have this catalytic cycle, which is dependent on the electrons from NADPH. So one of the things that both of these do is that the NADPH, this is a hydride transfer to reduce disulfides to sulfides. This is not really an electron transfer. Everybody kind of know what the difference is. These hydrides can transfer without you worrying about things like superoxide being formed or hydrogen peroxide. So again, this is a reduction of selenoproteins. So again, as I said, thioredoxin reductase is a selenoprotein here. And when you have thioredoxin here, it transfers the electrons through a hydride transfer here, and results in this selenium sulfide bond here, which then can easily be reduced back to the selenoprotein here, which then reconstitutes the active enzyme. So, is there any questions up to now? I know I'm just going through these as a category, but hopefully you'll have the slides and you'll come across these proteins and you'll be able to look it up. So, let's look at glutathione metabolism. Again, glutathione is really a very important front-end mechanism here. If glutathione is oxidized either by some powerful oxidants here, you can form this glutathione radical. The glutathione radical can rapidly react with another GSH to form this disulfide radical here. This disulfide radical here can be readily donate electron to oxygen to form superoxide and form GSSG. Well, that's fine because GSSG can be, by glutathione reductase, can actually reform GSH. So we have a cyclic mechanism. If we form superoxide, SOD, which we'll talk a little bit about later, superoxide dismutase, there are two forms. There's the manganese form, which is in the mitochondria, but there's also the cytosolic form, which is a copper-based, copper-zinc-based protein. And so that readily converts it to hydrogen peroxide. GPX here can convert it into water here. If GSH is in lower concentration, the thiol radical is in lower concentration, it can actually react with oxygen. And that oxygen, again, can produce superoxide, again, hydrogen peroxide H2O2, and form sulfenic acid, which sulfenic acid plus GSH goes to GSSG, and again, it can be readily reproduced to GSH. So there's a number of different proteins here that actually conspire to protect the cell from oxidation chemistry. And as I said, if you ever want to try something fun, treat your cells with BSO. You'll see your cells change dramatically just without even challenging. Their whole wiring, the system is not only in the toxicity, but once you deplete the glutathione, you've kind of depleted the insulation. So if you have ROSs coming out of the mitochondria, they're just going to come out. The glutathione and all the systems there that would normally abate them and isolate them into a certain area for signaling, they start going everywhere. So the cell gets very confused. It's kind of like going into a electrical box and kind of pulling wires out and taking all the insulation off the wires. Once you do that, then you're going to get a short. And that's what happens if you don't have glutathione. So there's some other things. We talked about this electron potential here. And if we look at some of these 2 electron reductions, this is dithiol DTT. Everybody's used this probably. in protein purification, or you get a protein. I'll give you a hint too. I've got to go quickly. But if I can give you anything useful, if you're working with proteins and you're actually looking at redox, be careful that they don't have a live DTT or some of the thiols in there. They're very powerful scavengers, and you'll treat with under some conditions, and all of a sudden it won't work. It's because the manufacturer has put DTT in there, and it's a very good scavenger of any type of redox. Lipoic acid, that's another one, thioredoxin. And so I gave this list here so that you have the kind of electron potentials, so you can actually follow the flow of electrons. In the case of nitrosative stress, this is a lot we've worked on here. When NO reacts with oxygen, you form GSH here. You form GSNO. Now, GSNO, I don't have it on here, is handled by a number of different mechanisms here. One that we showed was with copper zinc SOD. This is probably one of the primary reactions that actually can form these disulfide bonds here. It can actually transfer an NO here. NO can react with another GSH to generate HNO. This HNO can actually then be processed into this with excess GSH here, the sulfenic acid here. Sulfonamide here will form GSSG and GSH. So this becomes a detoxification mechanism for nitrosative stress here. Also, alcohol dehydrogenase 3 and 5, they actually will take GSNO and convert it into these products here, hydroxylamine and GSH. So there's a GSH, as we showed a long time ago, was actually critical in controlling the nitrosative stress as well as the oxidative stress. Ascorbic acid, everybody's heard about ascorbic acid here. This is the structure here. If you oxidize it, you form this radical formation here. When you take it, when you oxidize it here, you form the radical here. The radical has various different pHs here. These can take on, again, different types of electrons. And eventually, when you actually end up with DHA. And DHA is one of the primary products of the oxidation of ascorbic acid. And one thing I want to note is that vitamin C, or ascorbic acid, is usually about 0.2 millimolar in human plasma. Glutathione and ascorbic pools can actually communicate through things like protein disulfide isomerase and glutaredoxin, which is in the cytoplasm here, where you can actually get the conversion here of GSH to GSSG and vice versa here. And the electrons can be transferred here to things like DHA to form ascorbic acid, which can actually reduce metals here, or it can actually undergo oxidation potential here, and you get the protein, again, you can communicate between GSH and ascorbic acid. Uric acid, that's one of the products from the xanthine oxidase reaction. This is also a powerful oxidant, antioxidant, like ascorbic acid is. Tocopherol, which is vitamin E, and this is the structure here. And again, in the conjunction, you can have these highly oxidizing lipids. They can form a much more benign tocopherol radical, which then can be reduced by ascorbic acid here, and you get the reduced tocopherol. And this actually, reaction with tocopherol and ascorbic acid plays a critical role in detoxifying many lipid-type radicals. Some of the interactions of NO and ROS, as you say, nitric oxide combined to heme proteins. And what that does is that can generate HO1, and HO1 can generate CO here. Nitric oxide can abate nitric oxide synthase here. So a lot of times when we talk about oxidative stress versus nitrosative stress, they tend to be orthogonal because the chemistry of nitric oxide abates ROS chemistry and vice versa, and NO tends to prevent the assembly of things like NADPH oxidase. NO also binds to ferrochelatase. Some recent data showed that nitric oxide from heme also can prevent heme insertion. So there's all these feedback mechanisms here. NO can induce things like GPX and increase the antioxidant effects. And one of the things that I want to leave you with is that all the things that we've talked about here is that all these enzymes are dependent on one thing, oxygen. And if you ever ask, and people ask, why do we care about redox? Well, not only all the things that it does, but the fact is that we utilize oxygen. You have to consider all these redox intermediates. And from now on, I think, I've tried to lay out the chemistry and the biochemistry for you. Now we're going to talk more of the fun stuff about the physiology and signal transduction. But I wanted to give you at least a nomenclature of where things come from. So you got your freshman chemistry out of the way in the last 2 days. And so I think I'm going to hand it over to Terry now. What? Oh, you want that?
Okay, so Dave will be happy to answer questions at the end of the lecture. And now we're going to move on to David Roberts. And Dave got his PhD at the University of Michigan, and then he did some postdoctoral training there, came to the NIDDK, and subsequently he was hired by NCI. He's the chief of the biochemical pathology section. His title today, Regulation of Angiogenesis and Tissue Perfusion by Redox Signaling. Dave.
Okay, can everyone hear me? Forward. Okay. So you've heard all these enzymes and different RS and RNS. I'm going to be focusing on physiology, as Dave alluded, and I'll be focusing specifically on the cardiovascular system. So I'll put some emphasis on angiogenesis, but all of these pathways have dual roles in the cardiovascular system in terms of the genesis of a cardiovascular system and its regulation under physiological conditions, which controls tissue perfusion. So we're going to begin with defining what angiogenic factors are and angiogenesis inhibitors, how redox biochemistry fits into this pathway, and then I'll be talking about regulators of this system. So Dave has shown you a lot about the chemical and biochemical regulation, but at a higher level, there has to be systemic regulation of these processes. And so I'll be introducing one molecule, which is an angiogenesis inhibitor, which controls nitric oxide signaling, and then discuss some pathological conditions that are involved in the dysregulation of this pathway and how these can be targeted therapeutically in the cardiovascular diseases. And because I'm at the Cancer Institute, we're also interested in radiation as a therapeutic and how to exploit that for therapy. So beginning with a couple of definitions, vasculogenesis is the formation of new blood vessels. This happens in the early embryo when you go from having a completely avascular embryo to having its first vascular beds, which are formed out of mesenchymal cells that give rise to the first vessels. In the adult, you can also have vasculogenesis that occurs by bone marrow stem cells coming out into the periphery and forming new vessels. Once you have a primitive vascular bed, that has to differentiate into arteries and veins, so you have a complete circulatory system. And that process is known as arteriogenesis, which defines the direction of blood flow in a cycle. Then from this initial vascular tree, you can have branching that forms new vessels, and this is also important for disease, wound healing in the adult, and also in cancer. You form new vessels from branching from existing vessels. This is termed angiogenesis. And to complement the vascular tree, I'll show you in the next slide, we also have to have a lymphatic system to return fluid back to the central circulation. And the process that forms the second vascular tree is called lymphangiogenesis. Now, in normal circulation, you have blood vessels that carry red blood cells and white blood cells through circulation. These deliver oxygen and nutrients to the periphery, but they also are under pressure. And because they're under pressure and they are always leaky, at least at the molecular level, you have a continuous movement of water and electrolytes across the vascular walls, especially in capillaries. And so this fluid would accumulate in the periphery and you would have edema. But in healthy tissue, this is balanced because this fluid then moves into the lymphatics, which bring the fluid and other materials back to the central circulation. And so we have two parallel vascular trees, that carries the blood cells and that carries lymphatic fluid. And these are regulated by the formation of these, and their physiology is regulated by different factors. The formation of blood vessels and their maintenance is controlled by vascular endothelial growth factor as the primary driver. VEGF is a member of a family of vascular endothelial growth factors, and these act through VEGF receptor 2 and 1 in vascular cells and is the main driver of formation of new blood vessels. Once they're formed, they're not stable unless you have a parallel signaling system which comes from the angiopoietins. These signal through Ti receptors, and these together give rise to the formation of stable vessels. Lymphatics use another member of the VEGF receptor family and two different members of the VEGF family to drive formation of lymphatic vessels. Now, although I focused on the VEGFs, there also are other types of angiogenic factors. People who study developmental biology have focused a lot on VEGFs because if you eliminate any of these or even create a heterozygous mutant, you have a lethal embryo failure. So this is really the critical angiogenic factor. But in the adult and for other aspects of angiogenesis, you have a number of additional angiogenic factors. And in some tumors, you have other peptide mediators like adrenomedullin in breast cancer, for example, that can be the major driver of angiogenesis. And so even when you try to use drugs to block VEGF, you can still have angiogenesis that's driven through these other factors. And in vascular biology, we have this concept of an angiogenic balance. So when you're developing new tissues in the embryo, when you're repairing wounds in the menstrual cycle, you have formation of new vascularized beds, and so this side of the balance is dominant. But in a normal adult, in your tissues that are not proliferating, you have all of these positive signals opposed by anti-angiogenic factors. And so the normal physiology of the vascular system, and whether you're forming new vessels or whether you're causing regression of vascular beds, is all based on where this balance exists. In the context of cancer, we know that the growth of tumors beyond a certain dimension, a couple of millimeters, requires growth of new blood vessels. And so there's been much interest over the past 2 decades in developing angiogenesis inhibitors. And every time I give this talk, the list is growing. There now are a number of antagonists of VEGF. Avastin is an antibody that binds directly and sequesters VEGF. It was approved in 2004 for treating colon cancer, and now the list is continuously growing of the number of cancers for which Avastin, or bezacitabine as the generic, is being used to control therapeutic angiogenesis. There are advantages and disadvantages of Avastin, and consequently other companies are trying to develop other types of VEGF antagonists using siRNAs, using inhibitors of the kinases rather than of VEGF itself. These are in advanced phase trials, and there are other types of decoys that are being used, and some mixed kinase inhibitors that inhibit VEGF receptor and other kinases. And some of these have been approved for use in cancer. You can also go downstream, as we'll talk in a minute, from the VEGF receptor and inhibit targets of it inside the cell, inhibit integrins that mediate the adhesion of vascular cells to their matrix, steroids, cytokines, and also mimetics of the anti-angiogenic pathways that we're going to be talking about. So to understand why these work and why they fail, it's important to understand that the signaling systems that are being controlled, partially through redox signaling, are shared not just for angiogenesis, but also for controlling blood vessel tone and tissue perfusion. And so in this review we wrote a few years ago, we summarized some of the side effects that have appeared. All of these are drugs that were designed to inhibit angiogenesis. But curiously, one of the most common side effects of all of these drugs is that they cause hypertension. So we'll come into how redox signaling can explain why, when you're trying to inhibit angiogenesis, you're also inhibiting vessel tone and causing hypertension. So that leads us to the question of how does redox signaling fit into this angiogenic balance? And so we'll be discussing direct effects on angiogenesis, how redox signaling is involved upstream and downstream in controlling angiogenic factors, and then turning to the angiogenic inhibitor side and looking at the same questions. Now, this is a generic slide that applies to VEGF receptors, but also to other tyrosine kinase receptors, including that for PDGF and FGF that are involved in the various vascular functions. So all of these receptors, are tyrosine kinase receptors. They activate AKT and other kinases downstream that phosphorylate and activate, for example, eNOS in endothelial cells. And so activating these receptors leads to activation of eNOS, which increases synthesis of nitric oxide. They also regulate NADPH oxidases that you heard about in the last hour, and this leads to a cross-regulation between nitric oxide and superoxide. But it's also important that this superoxide is converted to hydrogen peroxide, and I'll comment back in more detail in a later slide to how that controls how long these tyrosine kinases remain active, because they're normally turned off by tyrosine phosphatases that remove the phosphate. But this feedback loop is very important for sustaining this positive signal by temporarily inactivating the phosphatases. These receptors also can lead to induction of hemoxygenase, which can produce carbon monoxide, and to enzymes that synthesize hydrogen peroxide. And so when we look at angiogenic signaling, it's really the integrated function of all of these different redox pathways that's important for understanding the physiology. So focusing first on nitric oxide. Dave mentioned in the last hour that at different concentrations of nitric oxide you have different chemistry, and that at low to moderate, so picomolar to low nanomolar concentrations of nitric oxide, which are typically produced by eNOS, this is the level of nitric oxide which is sensed by the heme moiety on soluble guanylate cyclase. And soluble guanylate cyclase, when it binds nitric oxide, turns on its synthesis of cyclic GMP. That cyclic GMP can then act on cyclic GMP sensors in the cell, leading to proangiogenic signals. They've also mentioned prolohydroxylase can be regulated by nitric oxide, and this in turn controls the stability of HIF1, and HIF1, among the genes that it controls, is the gene that encodes VEGF. And so if you have hypoxia, which would mean you lack the oxygen for this prolohydroxylase, or you have nitric oxide, you can control the stability of HIF1, and so under hypoxic conditions, you get induction of VEGF. So if there's chronic hypoxia, that says there are not enough blood vessels, not enough perfusion, so you want to turn on the synthesis of the biogenesis of more blood vessels to increase tissue perfusion. When nitric oxide concentrations are high, such as produced by pro-inflammatory conditions and you activate INOS, these levels of nitric oxide through other reactive nitrogen species can inhibit endothelial proliferation and angiogenesis, and also lead to induction of p53, which can lead to apoptosis and death of endothelial cells. So there's a bimodal function of nitric oxide on angiogenic signaling. The same thing applies to hydrogen peroxide. I don't want to go into this detail. In general, at low concentrations of hydrogen peroxide, as I mentioned, for controlling the tyrosine phosphatases, this is generally a supportive level of signaling for angiogenesis. But if you get to higher levels, then you start activating processes that inhibit angiogenic signaling. So to look at a little more detail in the tyrosine kinase feedback, I mentioned that we have tyrosine phosphatases. And in endothelial cells where VEGF is activating VEGF receptor, which becomes phosphorylated, this tyrosine phosphatase would be constitutively active and would only allow that to be active for a few minutes. But if you have signaling partially through RAC and partially through and other activities of NADPH oxidases, the hydrogen peroxide that's produced is able to oxidize sulfhydryls that are critical for the activity of these tyrosine phosphatases. When they become active, when they become oxidized, then they no longer can dephosphorylate. And then you have other pathways which reduce these back to the trial condition. And so, again, you have a way of turning off the pathway, but you can sustain it beyond what it would normally be able to do in the absence of this pathway. So there's really a cooperation here between nitric oxide signaling and peroxide signaling. So I also wanted to introduce hydrogen sulfide. This is a pathway which is attracting much more interest in recent years. Hydrogen sulfide is formed through, in mammals, generally formed from metabolism of sulfur-containing amino acids, cysteine, through three enzymes, cystathionine gamma lyase, beta synthase, and mercaptosulfotransferase. And all of these enzymes generate hydrogen sulfide. Hydrogen sulfide exists at neutral pH largely as HS-, but also with a substantial amount of hydrogen sulfide gas itself, which is then volatile. This circulates in plasma. Well, if you asked me 5 years ago when I started talking about this, we thought that it was 10 to 100 micromolar. But if you do the balance of how much sulfur-containing amino acids you ingest in a day, we don't eat enough sulfur-containing amino acids to make that much hydrogen sulfide. And so in reality, the current estimates are that the circulating levels in plasma, so I'm talking from the perspective of the cardiovascular system, it's normally seen high nanomolar to low micromolar levels of hydrogen sulfide. And this hydrogen sulfide is in pools. There's an acid labile sulfur, which is largely sulfur-bound to metals in proteins that are in circulation and in cells, and also to bound sulfane sulfur. And this is sulfur in the form of what's now becoming apparent as parasulfides, polysulfides, and polythionates and other structures. And these are all in reversible equilibria, so these both serve as buffers for the hydrogen sulfide that's in circulation. So even though you have this low micromolar, high nanomolar, there are also micromolar levels of iron, of acid labile sulfur, that are in equilibrium with this pool. And just like nitric oxide is degraded so that you limit its bioactivity, there also are important pathways that degrade hydrogen sulfide in mammalian cells. This is largely in the mitochondria and leads to oxidation to sulfite and thiosulfate. So just as we have a balance of nitric oxide signaling, we have a balance between the synthesis and degradation of hydrogen sulfide. And this is also a list that's rapidly growing. that a few years ago we didn't know any of these targets, but now there are a number of targets, and I put at the top of the list because we're talking about the cardiovascular system. VEGF receptor 2 is now a target of hydrogen sulfide. And so these are signaling targets. You heard in the last hour about all the biochemistry, but the biochemistry drives the modification of these signaling targets, and these in turn are driving the physiological functions. And you may hear in later lectures in this course about other functions of hydrogen sulfide in the CNS and inflammatory responses, but we'll just focus today on the cardiovascular system. So the first paper that showed that hydrogen sulfide has positive effects on angiogenesis was published now about 7 years ago. And they showed that if you look, and this is a classic matrigel assay where you put a matrix into a mouse and expose the mouse to different levels of hydrogen sulfide, you can see that there's an increase in the amount of hemoglobin in this matrix, which is indicative of how much blood is flowing through it, telling you how many vessels there are. We also showed in that same year that endothelial cell proliferation is driven by exogenous hydrogen peroxide, and that if we compare the responses that are induced by an NO donor and a hydrogen sulfide donor, we see similar increases in some of the downstream signaling, in this case, heat shock protein 27. Now, just last year, there was this nice paper that showed that one of the protein targets of hydrogen sulfide is VEGF receptor 2. And they identified these cysteines, 1045 and 1024, that are involved in a disulfide bond. But this can be either in the oxidized or the reduced form. And when this is in the reduced form, the kinase becomes active, and so we have proangiogenic signaling. When this is oxidized, all of these signals are shut off. So the ability of cells to respond to VEGF is modulated at the level of its receptor by hydrogen sulfide. And there can be multiple sources of hydrogen sulfide to drive angiogenesis. In the context of cancer, hydrogen sulfide, of course, is synthesized in endothelial cells, but in cancer, it's been seen that there's upregulation generally of the H2S biosynthetic enzymes. And so here's an example looking at HCT116 cells, that if you inhibit the H2S synthesis, you see similar results to inhibiting VEGF. And so the ability of the tumor cell to drive angiogenesis is in part mediated by hydrogen sulfide. In my lab, we've been looking at the synthesis of hydrogen sulfide also by T cells, which are important for regulating tumor growth, both by their immune modulating activity, but also by the hydrogen sulfide they produce. And what we showed was that when you activate T cells through the T cell receptor, two of the target genes of T cell activation are CSE and CBS. And so when T cells get activated, they're making more hydrogen sulfide. And so if you have CD8 T cells that kill the tumor, that's a good. But if you have other types of T cells which are not killing the tumor and they become activated, they can actually be producing hydrogen sulfide, which can drive angiogenesis in the tumor. So I'd like to go back to nitric oxide now. And the first take-home of this part is that nitric oxide is both upstream and downstream of VEGF. On the upstream side, we've already mentioned this, that nitric oxide, through controlling stability of HIF-alpha, drives the transcription of VEGF, which provides the ligand to signal through VEGFR2. And on the downstream side, VEGF has very redundant effects on nitric oxide signaling. So this is a complex slide, but I think the take-home message here is that this is a highly redundant positive effect on angiogenesis. So beginning here at VEGFR2, when it gets turned on, it activates a number of signaling cascades downstream of the receptor. One of these is the PI3 kinase pathway, which leads to phosphorylation of AKT. AKT is a kinase that phosphorylates eNOS on one of its serines, and this serine leads to phosphorylation, leads to activation of eNOS. You heard in the last half hour that eNOS also depends on calcium signaling. And VEGFR2, through phospholipase C-gamma, leads to increases in cytoplasmic calcium. That binds to calmodulin, gives a second activating signal. And just in case that's not enough, Sarc family kinases are also activated by VEGFR2. And there are phosphorylation sites both on eNOS at tyrosine 83 and on HSP90, and these further activate eNOS. So when VEGF binds to VEGFR2, you get very strong induction of nitric oxide signaling. eNOS then is active, produces NO, activates cyclic GMP, produces soluble guanylyl cyclase to produce cyclic GMP, and then this leads to various effectors that increase endothelial cell migration, vascular permeability, and growth, both through cyclic GMP and other pathways that are NO independent, which is part of the reason why an eNOS null endothelial cell can still respond to VEGF through these other pathways. So all of this is very highly redundant signal. So we've looked in a somewhat detail in how VEGF is tied into redox signaling. I don't have time to discuss all the other elements in this balance, but I wanted to point out generally that a majority of the proangiogenic factors regulate and or are regulated by redox signaling, mostly through nitric oxide. And as we're now going to turn for the remaining time, the angiogenesis inhibitors also are regulated by and regulate nitric oxide. And so this will show that the feedback regulation between angiogenesis stimulators and inhibitors is very intimately tied to redox signaling. My lab has been interested in thrombosponin 1, and this was identified in 1990 as an angiogenesis inhibitor. So we've known about its ability to limit VEGF and angiogenesis driven by other factors for a number of years. But I want to put this in a broader physiological context. So thrombosponin is a secreted protein. It's a trimer of very large subunits that have this sort of bola form, and this is reconstruction from crystals of various domains. Thrombosponin normally circulates in plasma. So it normally circulates at about 100 to 200 picomolar level. But in conditions of aging, diabetes, and injury, levels of thrombosponin go up, and in cancer, they go down. So why would we have this reciprocal regulation? Well, we found in 2005 that thrombosponin, well, we knew before 2005 that thrombosponin inhibits angiogenesis, but in most of the publications up until that year, it would require, in a pure cell culture system, about 10 nanomolar of thrombosponin to inhibit an angiogenic response. But I told you in the last slide that there's only about 200 picomolar of thrombosponin in circulation. So that would lead you to question whether this is really a physiological effect on angiogenesis. But what we found was missing in the assays that we did up until that point was nitric oxide. Because if you don't provide a way of synthesizing nitric oxide, it requires this high level of thrombosponin to inhibit. But when you're inducing NO synthesis, either by exogenous NO donors or endogenous synthesis, the ability of thrombosponin to inhibit shifts 3 log units to the left. So it becomes now, at physiological concentrations, able to inhibit nitric oxide signaling in endothelial cells. And through collaboration with the Link Lab, we also showed that nitric oxide at these physiological concentrations is able to control the synthesis of thrombosponin, which provides a feedback loop. So if we go back to the VEGF receptor signaling cascade, looking at this mechanistically, we found that thrombospondin binds to two receptors on the surface of endothelial cells. CD47 is the major receptor, and in cells that lack CD47, all this inhibitory signaling is lost. This signaling inhibits the activation of eNOS, it inhibits the activation of soluble guanylate cyclase, and it inhibits cyclic GMP-dependent activation of the protein kinase. So just as VEGF is a redundant activator of this cascade, the thrombospondin signaling is a redundant inhibitor of the same cascade. There's a second receptor that plays additional roles, partially by controlling the uptake of short-chain fatty acids, which are a further way of activating eNOS, which has to be lipidated to associate with the plasma membrane. But its activity genetically also relies on the presence of CD47. And then a few years ago, we found that there's an additional level of control at the level of the VEGF receptor. So CD47, it turns out, is constitutively bound to VEGF receptor 2. And by FRET studies, we can show that this is a very intimate binding between these proteins. So this is normally bound in a complex, but this is the inactive form of VEGFR2. When thrombospondin binds, this complex remains intact, but when VEGF binds to this, then this dissociates. And this dissociated complex, when it binds VEGF, no longer can be activated as a tyrosine kinase. So this provides another mechanism by which thrombospondin, by causing the dissociation of CD47 from VEGFR2, is able not only to control this downstream signaling at the level of eNOS and guanylate cyclase and cyclic G kinases, but also controls the upstream signaling, which is, as I've shown you before, through NO, but also through NO-independent pathways. And to generalize this further, a number of drugs, as I mentioned, have been developed for inhibiting VEGF signaling. And so these include the bevacizumab and the tyrosine kinase inhibitors. And these inhibitors also are able to manipulate nitric oxide levels, because if you prevent activation of VEGFR2, you decrease the activation of eNOS, so you decrease the levels of nitric oxide. And going back to the slide where I showed you the major side effects of these drugs were meant to inhibit angiogenesis. So it's good that they inhibit this signaling. But if they're inhibiting NO synthesis, the other physiological function of nitric oxide produced in endothelial cells is to control vascular smooth muscle tone, which controls perfusion. And so blood pressure and tissue perfusion are also controlled by these drugs that were designed to inhibit angiogenesis. If they shut down nitric oxide, you end up with hypertension by preventing the vasodilation. And so, and also there's a prothrombotic activity of these inhibitors. And so the side effects are actually understandable if you realize how they're signaling through redox pathways. Coming back to nitric oxide briefly, we have also shown recently that thrombospondin through CD47, because it's able to inhibit the synthesis expression of CBS and CSE, thrombospondin decreases the synthesis of hydrogen sulfide. And so in addition to its effects of the nitric oxide pathway, it's also inhibiting the pro-angiogenic activities of hydrogen sulfide and their immunomodulator activities. So coming back to the slide where we looked at the concentrations of thrombospondin, the normal physiological concentration in plasma of about 200 nanomolar is enough to tonically regulate nitric oxide and hydrogen sulfide signaling. In cancer, if you turn off thrombospondin, you're removing an angiogenesis inhibitor, but you're also removing a regulator of nitric oxide signaling, which can control perfusion in the tumors, as we'll discuss. discuss in upcoming slides. Conversely, in all the diseases of aging that we face where thrombosponin levels increase, these conditions all can lead to a complete shutdown of nitric oxide signaling. And so part of the pathology of diabetes, of ischemia reperfusion injuries, is through this ablation of nitric oxide signaling. So we've been looking at this in the context of ischemic injuries to develop therapies. How can we exploit what we've learned about this pathway to learn how to control this? And so an ischemic injury is where you have hypoxia, so you don't have enough perfusion and you want to restore perfusion, which can be driven either by increasing angiogenesis or by remodeling the existing vasculature so that you increase blood flow. And other studies have shown that nitric oxide is intimately involved in overcoming ischemic injuries. So the model we use is called a McFarland skin flap, where you make an excision on the back of a mouse. This creates a flap where at this end there's good perfusion, and as you go down the flap there's increasingly more ischemia. And so in a wild-type mouse, the distal region of this becomes necrotic because it doesn't have enough blood flow. But in a thrombospondin null mouse, so we've taken this inhibitor of nitric oxide signaling away, we saw that these mice were able to completely maintain viability of this ischemic tissue. This was also seen by ligating the femoral artery, which eliminates circulation in the hind limb of these mice. And in a wild type, or CD47 null, lacking the thrombospondin receptor, at day 0, there's no circulation in this hind limb, but 7 days later, it's been restored only in the null. So taking away this inhibitory pathway has restored this, and also in skin grafts, being either null for thrombospondin or CD47 overcomes this extreme ischemic injury. So in this context, it's good to be null, but how can we exploit this? Well, first I want to put this in the context of how do we integrate angiogenesis with other vascular physiology. Nitric oxide is important in several aspects of circulation. In platelets, if you cut yourself, you need nitric oxide signaling to be limited in order for platelets to activate and clot. Nitric oxide is normally a tonic inhibitor of hemostasis. And this happens on a time scale of seconds to minutes. So nitric oxide signaling, as Dave mentioned, tends to not travel very far in circulation, acts very locally in hemostasis. Vasodilation happens over a longer time scale, minutes, seconds to minutes to hours. And angiogenesis, the ability to generate new vessels, that takes days. So that's a long-term response. So we've looked at acute changes in blood flow and found that if you inject a nitric oxide donor into mice, this normally leads to a transient increase in blood flow by vasodilation. This can be followed by imaging. And so here's a functional MRI where we measure the increase in oxygen concentrations in tissue. And we see that in a wild-type mouse, if you inject an NO donor in the tail vein, I'm sorry, this was rectal actually, you see an increase in oxygenation in the blood that happens within a few minutes. But if you lack thrombospondin, you get more of an increase. So this is on the scale of seconds to minutes. We can increase perfusion by eliminating thrombospondin. And you can see this also by laser Doppler imaging in the ischemic injury. If we make an ischemic injury and look over now a scale of minutes, we see that in a wild-type mouse, you have a rapid loss of oxygenation, of perfusion rather, in the injured tissue, and less so in the thrombospondin null. So the absence of thrombospondin has acute effects on how blood flows. Conversely, in a tumor, we see the opposite. So here we have a tumor on the leg of a mouse, and this is the control limb. And when we measure the oxygen and the blood flow through the tumor and versus the normal limb and challenge the mouse with a nitric oxide donor, we see that in the control limb, just as in the normal limb I showed you in the previous slides, we have an increase in perfusion. But in the tumor, you have a decrease. And the explanation for that is that the blood vessels in tumors tend to not be regulated normally. They can't dilate and constrict in response to vasodilators and vasoconstrictors as well as normal vessels. So if you have an artery that's supplying both the tumor and other adjacent tissue, and you have balanced flow between these, if you use an NO donor, it will dilate preferentially the vessels that go around the tumor, and you end up with less blood flow going through. Conversely, if you use a vasoconstrictor, such as an adrenergic agent, you constrict selectively the vessels that go around the tumor, and you end up with more flow to the tumor. And so this is collectively known as the Steele effect. Let's skip this. Fine. So, I've shown you in cancer and in other injuries where changes in NO signaling can lead to abnormal growth of vessels and perfusion, which you want to limit in a cancer. And in ischemic tissues, you have a deficiency of angiogenesis and perfusion that you want to increase. And so if this pathway is controlling that, how can we manipulate this? So we can use antibodies to prevent thrombosponin from binding to CD47. We could add antibodies to CD47 that prevent thrombosponin binding. One approach that we found very useful is to temporarily suppress the expression of CD47. And for that, we use antisense oligonucleotides known as morpholinos, which is a very stable type of antisense. And we can use that to block the translation of the messenger RNA, which then leads to decreases in CD47 expression. So the context where we want to increase NO signaling includes conditions such as reconstructive surgery, ischemic injuries, and other chronic diseases that could create chronic conditions of ischemia. And we would like to increase this inhibitory pathway in the case of cancer. And again, because we have limited time, I'm not going to go through this. You have these in your handouts. You're welcome to read these papers. We've shown in the ischemic injuries that if we use these approaches, we can protect tissues in normal wild-type mice from ischemic injuries. We've extended this to pigs and shown it both for fixed ischemic injuries and also for full-thickness skin grafts. We've shown that we can protect aging mice from loss of the ability to recover from ischemic injuries by blocking this pathway. This also extends to reperfusion injuries, which are somewhat more complicated and involve other pathways in addition to nitric oxide. And recently, two interesting papers have come out in the past few years showing that kidney transplantation, where you get a donor kidney either from a live donor or from a cadaver, it has to be flown on an airplane to the recipient. And during the time that it's sitting outside of the body, it becomes progressively ischemic. And when it gets connected back into the circulation of the recipient, it undergoes a reperfusion injury. And these papers show nicely that blocking CD47 allows you to prevent that reperfusion injury in kidney transplantation, both in mice and in rats. I think I can skip this too. So I wanted to tell you more about cancer, because that's what pays my salary. And we keep being told we have to stay on mission. So blocking this pathway, I've shown you, has effects on angiogenesis. It has effects on ischemic injuries and reperfusion injuries. So we wondered whether the stress, whether this generally applies to stress rather than just these specific vascular injuries. And the first clue to that was when we irradiated the hind limbs of our mice. So a wild-type mouse, if you give it a dose of radiation, 25 gray, which is actually less than the cumulative dose a cancer patient will receive to try to cure a tumor, this causes a lot of damage. You lose your hair, as cancer patients know very well. They can get ulceration in the skin, known as desquamation. And if you look 2 months out after radiation, there also is progressive fibrosis of the irradiated tissue. And you can see that by the retraction of the limb. The limb is drawn up in the wild-type mouse. But in our null mice, we saw that there's less hair loss, almost none in the CD47 null, and the limb remained flexible. And if you look at histology from these tissues, the skin histology is kind of obvious from looking at the growth picture here. All the hair follicles are gone in the wild type. Some are maintained in the CD-respondent null quite well in the CD47 null. But what you can't see here from the outside is the muscle pathology. So these light area are all muscle fibers that have died after radiation and is resulting in the fibrosis. And this is essentially absent in the null animals. Fortunately, to study the signaling, we could reproduce this in isolated cells. So this radioprotection is cell autonomous. If we take primary cells from the lungs of our null mice, they are very resistant to radiation, whereas wild-type cells die progressively with increasing radiation doses. And if we use human cells and treat them with our morpholino that suppresses CD47, this protects them from the damage caused by radiation. Now, is this caused by nitric oxide signaling? There are papers, including from our previous speaker, showing that nitric oxide has radioprotective activity. But what we found was that if we block NO synthesis, we can still protect these cells by knocking down CD47. And conversely, that adding an NO donor is not sufficient to get radioprotection in the absence of blocking this pathway. So, or adding a cyclic GMP donor. So it's more than just the NO cyclic GMP pathway. And again, I don't have time to talk about the mechanism. You're welcome to look at this paper. We showed that it's autophagy, which is a protective response of cells to stress, which is regulated by blocking CD47, which upregulates the expression of a number of elements in the autophagy pathway. And if you use an siRNA to block the autophagy pathway, you block this protection. So this is critical for that. And we can extend this to total body radiation, which if you can't get money for cancer, you can go to the DOD. They have a lot of money, at least I'm told. And so they're interested in having troops and civilians survive total body radiation. And we were able to show that if we block CD47 in mice exposed to, in this case, I think it was 7.6 gray, so basically an LD70 dose for these mice, we both increased autophagy responses in them and increased their survival. So, adding on to the pathways that I've shown you downstream of CD47, we still have the NO cyclic GMP signaling pathways, calcium signaling, regulation of proangiogenic signaling upstream through VEGFR2. But the new branch is that this also controls the expression of Becklin and downstream effectors of autophagy. Other studies have shown that CD47 also controls the localization of the NIP3, which is a protein that causes mitochondria to depolarize, release cytochrome C, and make a lot of ROS, as you heard in the last hour. So, to conclude, give you time for a few questions. The second half of the lecture today has focused on an inhibitor of redox signaling, thrombespondin. It's a physiological and pathophysiological inhibitor of nitric oxide signaling, hydrogen sulfide signaling. These limit in endothelial cells angiogenesis. In smooth muscle cells, it regulates smooth muscle tone, which controls perfusion of tissues. and it controls cardiovascular dynamics. This is largely mediated through a receptor, CD47, which does the opposite of what VEGF does to nitric oxide and hydrogen sulfide signal. VEGF turns it on in vascular cells, CD47 turns these pathways off. And so in the cases of pathology such as ischemia reperfusion, and radiation, these turn off nitric oxide signaling. In the case of ischemia, simply turning on the NO-cyclic GMP pathway restores function and can overcome ischemic injury, but for radioprotection, you need a second pathway, which is independent of the redox signaling, and that we're on the way to developing drugs targeting this that can improve nitric oxide signaling under ischemia and aging so that we can all live forever. We want to do that. And in the case of treating cancer, that we can modulate the radiosensitivity of healthy tissues. So I will stop there, and I guess we'll both answer questions.